Cybersickness exploration for different VR tasks under variable rendering conditions. In virtual reality, cybersickness can seriously affect user experience. A comprehensive understanding of this phenomenon is essential for enhancing the user experience in VR. Current studies have focused on how factors such as user speed, field of view, frame rate, and optical flow affect cybersickness. However, there is limited research on the effects of basic VR tasks under different rendering conditions. In addition to simple browsing tasks, in real-world VR application scenarios, which often include a wider range of complex tasks such as selection tasks and manipulation tasks. Notably, rendering directly influences the visual quality and realism of VR content, making it a potential factor in cybersickness. Therefore, a thorough investigation of the interactions between different VR tasks, rendering conditions, and cybersickness is necessary. We designed a user study investigating cybersickness for different VR tasks under variable rendering conditions. A is the experiential setup, including the VR tasks and rendering conditions used in the experiment. B is the data capture part, showing the equipment and devices including HTC Vive, the EEG device, and the monitor for playback. C is the process of processing and analyzing subjective and objective measurements. In this study, the VR environment was designed as a laboratory with lab objects such as microscopes and test tubes. We asked participants to complete the task of browsing, selection, and manipulation tasks. In the browsing task, participants were asked to follow a given path to navigate and browse the scene along the way. In the selection task, participants were asked to choose 4 to 8 specified laboratory objects. In the manipulation task, participants were asked to place 4 to 8 laboratory objects into a specified target location, which required them to translate, rotate, and adjust the objects as needed. 2 key rendering conditions in VR are investigated. Central area radius controls the radius of the high-resolution field of view at the center of the user's VR view. Peripheral resolution defines the resolution of the peripheral regions outside the central area. A total of 24 participants were enlisted to finish the VR tasks under varying rendering conditions. The entire process video and EEG signal were recorded simultaneously. The experiment consists of 3 CAR settings, 3 PR settings, and a baseline setting, a total of 30 task trails. The whole experiment takes about 45 minutes to complete. We group 3 trials of different VR tasks into a session. The figure illustrates the procedure for one session. Each session begins with a 30-second positive mindfulness to establish a baseline for the EEG signal. Participants then began the first VR tasks trail and reported cybersickness. A 15-second rest was taken between trials to facilitate recovery. Participants then began the second and third trials and reported cybersickness. After this, participants watched the recorded video, paused the reported moment, recalled their experiences, and filled out the SSQ. HTC Vive HMDs were used to provide VR task in this study. EEG signals was captured by using the Smarting Pro 32 EEG device of mBraintrain and was used as the objective measurement in this study. The moments of cybersickness was reported by pressing the button on the controller. The severity of cybersickness was obtained by using the SSQ and used as the subjective measurement in this study. We analyzed changes in the PSD of EEG signals in the alpha and theta bands under conditions without and with cybersickness, as shown in the figure. We conducted paired t-tests on each frequency band in the parietal and occipital lobes. The results showed significant differences in the alpha band. We further investigated whether PSD values were associated with subjective cybersickness severity by conducting a correlation analysis between PSD and SSQ scores. The Pearson correlation coefficients between PSD values and SSQ scores across the alpha and theta frequency bands and the parietal lobe and occipital lobe as shown in the table. The results indicate moderate positive correlations between PSD values and SSQ scores for both the alpha and theta bands in the parietal and occipital regions during cybersickness. Notably, the alpha band PSD in the parietal lobe exhibited the highest correlation with SSQ scores. We examined how task types affect cybersickness severity using the SSQ and alpha band PSD. The results are shown in the figures. We found a significant main effect with browsing tasks inducing the most severe cyber sickness, followed by selection and manipulation tasks. These results suggest that task type may influence cyber sickness severity, with the browsing task associated with higher symptom scores compared to the other task. This figure illustrates bar charts depicting the SSQ scores and PSD values under various CAR and PR settings. Our findings indicate that elevated CAR settings potentially induce more severe cyber sickness, as consistently reflected in both SSQ scores and PSD results. Conversely, PR demonstrates little impact on cyber sickness. This figure presents 6 representative time frequency analysis results from our experiment. The rows correspond to signals from the parietal and occipital lobes. The columns represent different VR tasks. A prominent red vertical line at x equals 0 marks the moment of user-reported cybersickness. The black dots denote points where a PSD increase was detected, aligning with the first purple vertical line. Black dashed lines indicate the corresponding latency L, indicating the latency of brain activity associated with cybersickness. These results indicate a measurable time lag between cybersickness-related EEG signals and the reported onset of cybersickness. This figure shows cybersickness frequency across different task types and rendering conditions. Results indicate that the browsing task induces the highest frequency of cybersickness, followed by the manipulation